Yep. Thanks for the opportunity. An unintended life's work later led to this paper last year where we found a new gene variant that appears to only be existing among Pacific peoples, and that's because of human evolutionary history and probably because of, we believe, natural selection. That gene variant is positively associated with obesity, but inversely associated with type 2 diabetes. So this got a fair amount of press, it got in a good journal, and our future work and our current work, which is summarized here with a lot of words, which I'll try to walk you through, is basically aimed at figuring out what in the hell is going on with the physiology of this gene and the behavior related to this gene. How does it actually work? How is it that we see this somewhat paradoxical relationship? And building on that we'll get to is, are there potential for us to understand what these mechanisms are that would allow us to build interventions that would be genotype specific interventions? Say what you will about the increasing emphasis on genomics as perhaps misplaced. The scientific questions remain that if we can customize interventions based on people's individual inheritances, both behavioral and biological, that's probably a scientific advance that we believe is important for clinical medicine, but actually even really for public health as well. So we have a current grant that is ongoing right now. We have people in the field. Courtney Choi, who's a new PhD student, was in the field in Samoa with some of the people who are there now. This is the variant, and so we're trying to understand what exactly its impact is on metabolic and behavioral traits, particularly those affecting energy homeostasis. And we're choosing people whom we had seen before in our genome-wide association study, GWAS, and we're doing more intensive phenotyping. So we're doing an actual oral glucose tolerance test with those people, a research quality one, so it means as opposed to a clinical one, that we're taking more frequent blood samples to really understand the disposal of glucose and the activation of insulin because of the glucose load that they take. And we're spending about $100,000 of your taxpayers' money getting a DEXA scanner to Samoa. It's there, it's working. So because we're trying to understand where is the adipose tissue, we also have opportunities to ask other questions about bone mineralization and even some potential cardiovascular scanning with the model of the scanner we're doing. But really what it looks at is body composition, because it may be that this gene, though it contributes to obesity and appears to be protective against type 2 diabetes, one potential mechanism is it puts adipose tissue in less risky places, which of course relates to stem cells that become pre-adipocytes, that become adipocytes. So we're looking at a lot of these things in some physiological detail. We're hoping to get adipose tissue samples from these individuals so that our fat cell biologists can look at those fat cells in terms of genetic messaging, in terms of messenger RNA and gene expression to try to understand what might be going on. Because the obesity field is riven right now by divisions between whether obesity is sort of centrally mediated or perhaps fat cell mediated. Like everything, it's probably a little bit of both, and it depends on your family and your ethnicity. And so there's interesting questions to us. We're explicitly going to be doing G by E, gene by environment interactions, looking at this particular variant and how the effect of this variant modifies dietary impacts on, say, metabolic phenotypes, or including obesity, lipid and lipidemic glucose diabetes traits as well. physical activity, diet, smoking, a variety of things that we collected in the parent GWAS study that we ran out of money and time to look at. So that's a major area, and something that any master's trained epidemiology student could pursue. Because we could treat this variant as a simple categorical variable. There are some special genetic statistical techniques that we can do, but it's relatively straightforward. It's not as complicated as doing a GWAS analysis. And I want to mirror something that Greg said. Many of us have what could be politely called underexploited data sets. We spend a lot of money, we collect a lot of data, we get those first several papers out, because those are the ones that are priority or the ones that we think are hot or would get in a big status journal. And then we run out of time and money and personnel, and we have plenty of worthwhile, they're not B-list questions, they're still very, very important questions that graduate students are amply well trained to pursue, particularly with close mentoring. So that's an invitation if you can't figure that out. We're also going to be testing gene-by-gene interactions, because we have 900,000 other genotypes, and as a matter of fact, we're getting whole genome sequencing now done on these individuals because we're part of the US Top Med Consortium. So we're literally getting the AGTC throughout the entire genome for about 1,200 people from whom we will impute to the rest of the sample. And then we'll be looking for associations with these newly gathered phenotypes, the OGTT, the DEXA body composition, and also the behavioral traits which Professor Field helped me and my colleague Nicola Hawley at Yale, who's a partner in this, to begin to develop out in the field right now. We got a 7th percentile on an NIH grant. That's a good number for those of you who aren't. And so we're expecting this new grant to start sometime this fall. It will have new field work. We'll be piggybacking on this for some field efficiencies, hoping NIH doesn't think it's too efficient and cutting us too much. And basically we're going to be looking at energy metabolism, energy balance, and homeostasis using some of the energy expenditure methodologies. So we'll be looking at total daily energy expenditure using doubly labeled water, which many of you know is a tracer, a way to determine the primordial cellular energy and whole body energy expenditure. And on top of that, we'll be doing resting metabolic rate, the thermic effect of food, with indirect calorimetry, collecting gases and looking at expired gases for the respiratory quotient estimations. I'm actually a biological anthropologist who studied a lot of these things, so I'm really happy to be able to be doing these things in the field. And we'll be looking at measured energy intake. We've published a lot of things with the help of Anna Bailin, who was a faculty member here, is now at University of Michigan, a diet and nutritional epidemiologist, relying on reported intakes. And everybody knows that those have weaknesses, like reporting all the important things in life, like drinking and sex and physical activity, right? There's a lot of error related to that. And we're going to be using more objective measures to get at that, because we're really trying to understand, do people with this variant, because we're going to be choosing them by genotype again, have something different about their fundamental energy expenditure or energetic efficiencies. And we're going to be following these individuals over a 2 to 3 year time to see if we can actually detect changes in body weight and body fat, because we'll put them on that DEXA machine that we have out there to look at. In addition to that, we're going to be doing more maternal child health studies, particularly the things that Courtney is interested in, and the thing that Nikki at Yale, Nikki Hawley and I are interested in. And so there's a picture of our DEXA, there's a picture of Courtney in the field. This is Samoa. Ask me some questions. Come talk to me. Maybe you can go to Samoa if we can get the money right for you. Happy to take questions about